So, when you hear the name E. coli, what's the first thing that pops into your head? Is it a dangerous foodborne illness? Or is it the absolute workhorse of the modern biology lab? Well, the surprising answer is it's both. Today, we're going to dig into the fascinating and kind of weird dual identity of Escherichia coli.
So here's how we're going to break it down. First, we'll try to untangle this split personality. Then we'll get into the puzzle of its different pathogenic versions, follow a really surprising molecular clue, and then see how modern techniques let us basically read the bacteria's mind. After that, we'll see why it's so important to take these experiments out of the lab and into a live host before finally looking ahead at what's next in the fight against these infections.
All right, first things first, workhorse or pathogen. This really is a tale of two completely different bacteria, somehow living under the very same species name. Yeah, this image just captures that paradox perfectly, doesn't it? Over on the right, you've got the workhorse, this little microscopic factory we've engineered to make everything from biosensors to new biomaterials. And then on the left, you've got its dangerous alter ego, the pathogen. It's armed with these molecular weapons, like something called the type 3 secretion system, or T3SS for short, and toxins that can cause some really devastating diseases. This quote from Neidhart pretty much says it all. For nearly 100 years, E. coli has been biology's go-to model organism. Why? Well, it grows unbelievably fast, it's not picky about lab conditions, and its genetics are just a dream to work with. I mean, this is the organism we used to figure out the absolute fundamentals: DNA replication, transcription, translation, you name it. But here's the crucial thing to understand. The lab strain we all use, E. coli K12, is basically a domesticated version of its wild ancestors. Think about it. It's been living in labs since 1922, just passed from one culture tube to the next. It's been zapped with harsh mutagens to get rid of things like lambda-phage, and as a result, it's just not tough anymore. It's actually a really poor colonizer of the gut now. Its cozy life in a flask of rich media is absolutely nothing like the hostile world its relatives have to face out in nature.
And that brings us crashing back to the reality of its pathogenic side. This headline from that huge outbreak in Germany back in 2011 is, well, it's a chilling reminder. An E. coli strain, which they eventually traced to contaminated bean sprouts, caused thousands of illnesses, severe kidney damage, and dozens of deaths. This was no tame lab strain. This was a highly adapted, incredibly dangerous pathogen.
This huge difference between the lab bug and the killer bug brings up a really central question. And that brings us to our second section, the pathotype puzzle. How is it that different strains of the exact same bacterial species can cause such wildly different diseases? So this diagram here shows you just how diverse pathogenic E. coli, or pathotypes, can be. You've got MEC, that's neonatal meningitis E. coli, a major cause of that disease in newborns. Then there's UPEC, or uropathogenic E. coli. That's the main culprit behind urinary tract infections. And finally, the one we're going to focus on today, EHEC, or enterohemorrhagic E. coli. These are the strains like the infamous 0157 that are strict gut pathogens, setting up shop in the colon. And this specialization leads to a really cool scientific mystery. In the lab, in vitro, EHEC will happily stick to all sorts of human cells, lung cells, bladder cells, whatever. So the big question is, why? Why, in a real, live host, is it found almost exclusively in the large intestine? What is it about that specific environment that dictates this tissue tropism, this preference for one tissue over all others? The search for an answer to that question takes us to our third section, a D-serine discovery. The first big clue came from a really unexpected place, a simple and often ignored little molecule. Okay, but before we get to that molecule, we need to properly introduce EHEC's number one weapon. It's called the type 3 secretion system, the T3SS. And honestly, the best way to think about it is as a tiny molecular syringe that the bacterium builds on its own surface. It uses this thing to inject its own proteins, we call them effectors, straight into our host cells. And these effectors then basically hijack the cell, letting the bacterium attach and cause disease. And this is where the breakthrough happened. As biology students, you learn all about L-amino acids. They're the building blocks of proteins. But their mirror images, D-amino acids, they exist in nature too. Researchers tested what would happen if they exposed E. coli to various D-amino acids. And the result was, whoa. It was striking. So this chart is showing the expression of a key gene needed to build that T3SS syringe. Normally, expression is high, But when the bacteria are exposed to D-serine, look at that, expression just plummets. D-serine is acting like a signal, telling the bacteria to turn off its main weapon. Okay, but does turning down the genes actually stop the infection? Well, this chart shows you the real-world consequence. Here, they measured the percentage of host cells that EHEC could infect. In the control group, you can see infection rates are high. But when you add D-serine to the mix, the number of infected cells just drops off a cliff. So the key takeaway here is that this simple molecule doesn't just tweak gene expression. It functionally disarms the bacterium, at least in the lab.
This is a massive clue, right? But how? How does D-serine have such a huge, specific effect? To figure that out, we've got to look way beyond just one gene system, which brings us to Section 4, decoding the bacteria's response.
So, to get the full picture of what's going on inside that bacterial cell, we need a way to look at everything at once. The best analogy I can think of is imagining you could read all of a cell's internal memos at the same time. Every active gene makes an RNA message, a transcript, and with this incredibly powerful technique called RNA sequencing, or RNA-seq, we can grab and sequence all of those messages, all 5,500 of them for E. coli, simultaneously. It gives us a complete snapshot of everything the cell is doing. And what the RNA-seq data showed was this fascinating global shift in the bacteria's entire behavior. So first, it confirmed what we already suspected. The T3SS genes were shut down hard. But it also showed something totally new. D-serine was turning on the bacterial SOS response, which is a famous pathway for dealing with stress and DNA damage. And maybe the most exciting part, the data pointed to a gene that was basically unstudied, called YAJ, as a key master regulator that was controlling this whole network. Okay, so now we have this beautiful molecular mechanism all worked out in a petri dish. But the big question is still hanging there. Does any of this matter in a real infection? And that brings us to the most critical step in all of biomedical research, our fifth section, testing in a live host. This slide really just lays out the massive challenge of moving from the lab to a live host. On the left, you've got the in vitro world of the lab. It's simple, it's defined, the bacteria are everywhere, so getting a strong RNA signal is easy. But on the right, that's the in vivo world. A living host is unbelievably complex. You've got an immune system fighting back, other microbes, tons of host factors, and crucially, the bacteria you're studying are just a tiny fraction of all the cells, which makes their RNA signal incredibly hard to find. So how on earth do you study a human gut pathogen in vivo? The answer is to use a really good animal model. In this case, researchers use a bacterium called Citrobacter rodentium. It's a natural mouse pathogen that, and this is the critical part, uses the exact same T3SS-based mechanism to cause disease in the mouse gut that EHEC uses in us. This makes it the perfect stand-in for studying the fundamental process of infection.
And the way they do this experiment is just so elegant. First, they infect mice with Citrobacter. Then, over time, they isolate the infected gut tissue. The next step is to pull out all the RNA from that tissue, remembering that something like 99.9% of it is going to be from the mouse. The really tough technical bit is to specifically enrich for that tiny, tiny fraction of bacterial messenger RNA before finally running the RNA-seq to see what genes the pathogen has switched on during a real fight.
And the result of that incredibly challenging experiment? It was completely unambiguous. When you look at which genes the bacteria are cranking up the most while they're inside the host, one category just blows everything else away: virulence factors. They absolutely dominate. See, inside a host, the bacterium isn't acting like it does in a lab, where it's all about growing fast. No, it's behaving like a warrior, pouring all its energy and resources into making the weapons it needs to fight the host and win. This is such a powerful finding. It shows us how bacteria really behave during a disease. And that brings us to our final section, the next frontier in infection. How do we take this fundamental knowledge and turn it into something that can actually help people? So let's put all the pieces together. We've seen that specific things in the host, like the amino acid D-serine, can act as signals that bacteria use to control their weapons. We've also seen undeniable proof that pathogens play by a completely different rulebook inside a host versus in the lab. And what that means is if we want to develop effective new treatments, we have to study bacteria in the context of a real infection. Getting a deep understanding of these in vivo mechanisms is absolutely key. And this leaves us with one last big forward-looking question. We're living in an age of ever-increasing antibiotic resistance, where our old strategy of just trying to kill bacteria is starting to fail. But what if we could use these fundamental insights to design smarter anti-virulence therapies? Instead of launching a lethal attack, what if we could develop drugs that just disarm the pathogens? Drugs that could mimic host signals like desearing and essentially tell them to put their weapons down. This shift from killing to disarming, that's the challenge and the incredible promise for the next generation of infectious disease research.